Hello everyone, my name is Meredith Sanders and this is Lab 5 called Membrane Structure and Function for Intracell Bio 2440 with Dr. Hudson online. This is all my definitions basically broken up into sections. The first section is pink and it signifies the structure of the membranes. My second one is blue and it's for the characteristics of parts in the membranes. Then we have number 3, which is a green, and that's transporting and transforming. And then we finally have number 4, which are our models, and those are in purple. So just to quickly kind of go over these. Alright, so we start off, we have plasma membrane. Plasma membrane is just the membrane that separates the outside environment from the inside. So it's for protection and stuff for the cell. Phospholipid bilayer is an arrangement of lipids and phosphates that make a cell membrane in order to monitor traveling of molecules and ions that come in and out of the cell. Then we have electrochemical gradient, and that's just a gradient of electrochemical potential that lets ions move along and across the membrane. Then we have a gated ion channel, and that's a channel of cells that it only opens its gate, as we call it. It only opens its gate for ions to pass under certain instances. So it's kind of like a monitor, it's kind of like a gated community, you know? So then we have a concentration gradient, and that's where your solutes are moving from a solution with lots of particles to a solution with less particles. Then we have a membrane channels. That's channels that allow the movement of ions and water and other solutes to go through the membrane. We have integral membrane protein, which are the membrane proteins that attach to biological membranes permanently. That's the difference between integral membrane protein and peripheral. Integral is the proteins that attach to biological membranes permanently, but peripheral are the ones that only attach to the biological membranes temporarily, then they break off. Ion channel is just a protein that transverses the lipid bilayer of cell membranes to create a channel. We have osmosis, which is a type of diffusion where the substances, they cross the semi-permeable membrane, and they do that to balance the concentration of other substances. So it's kind of like comparing. Selective permeability is the property in cellular membranes where only certain molecules are allowed in and out of the cell. So it's like it says, it's selective on what is allowed to come in and out of the cell. Then we have vesicles, which are the structures that are inside or outside of the cell that's made up of like a liquid or cytoplasm, and that's enclosed. The vesicles are enclosed by a lipid bilayer. Then we have glycolipid, and glycolipid are lipids with carbohydrates that are attached with this covalent bond, and that creates stability for the cell membrane. And then we have a glycoprotein, which are just proteins with a sugar attached to it. So that covers the structures of the membrane. Then we're going into characteristics of like the parts that are in membranes, because it has many different characteristics. So we're starting off with a hydrophilic head of a phospholipid. That's the part of the phospholipid molecule that's made of an alcohol and glycerol group. And as it says, hydrophilic, that means that it likes water. So the head, the hydrophilic head of the phospholipid likes water, right? And then we also have a hydrophobic tail of the phospholipid, and that's the part of the phospholipid molecule that's made of fatty acids, and because it's hydrophobic, it dislikes water. We have a hypotonic solution, and that's just a solute concentration where it's lower than the comparing solution. Now the difference in contrast between hypotonic and hypertonic solution is the hypertonic solution is where the solute concentration is higher than the comparing solution. So hypotonic is lower than the comparing, hypertonic is higher than the comparing, and isotonic solution is where the solute concentration is the same as whatever solution you're comparing it to. Then we have a saturated fatty acid, and that's a fatty acid that doesn't have like unsaturated linkages between its carbon atoms. So the saturated fatty acid, it can't absorb more hydrogen. However, the unsaturated fatty acid, that's a fatty acid that has one or more double bonds, and so that means that the unsaturated fatty acid can absorb more hydrogen atoms. Then we have a proteoglycan, type, which is a type of macromolecule with high molecule weights, so the high molecular weight, and they are in the They're in the connective tissues of the body. Then we have sterols. It was put as sterols/steroids, but basically sterols are a form of steroids. And steroids are just a lipid. And they're kind of like how in animals, cholesterol is a sterol that's in animals. But steroids, they're actually just like a group of hormones with 4 rings of carbon atoms, and so if you see 4 rings of carbon atoms, and it's a group of hormones, we can say it's a steroid. So sterols are just a type of steroid. Then we have a detergent molecule, which is a molecule with polar and nonpolar hydrophobic domains. So detergent molecules have polar, likes water, and nonpolar hydrophobic domains. So doesn't like water and likes water. So it has 2 in 1. And amphipathic molecules are also molecules with both polar and nonpolar parts. But the detergent is like the domains, and the amphipathic is just, it has the different parts of it, not necessarily domain. Membrane proteins are a type of protein that is attached to a biological membrane, as we can tell. There's just a protein attached to the membrane. And then monolayer associated membrane protein is just that proteins that are situated inside the cell, the cytoplasm. Lipid-linked membrane-associated proteins are that lipid components anchor the protein to the membrane. So this is just saying that lipids, they're able to anchor the proteins in here, into the membrane. And then we have protein-attached membrane-associated protein. It's a mouthful, but it's just the saying that membrane proteins are basically the gatekeepers for the cell. They monitor what comes in and out. They open and close, and those are the different characteristics. As you can see, we talked about how things are hydrophilic, hydrophobic. They can have polar, non-polar parts. It's just describing all the different characteristics of the parts that are in membranes. Then we move on to the green, which is the transporting and transforming. And we start with passive transport, which is substances that are transporting along the gradient of concentration. And we call it passive transport because you don't need chemical energy to go downhill, because it's going from a high to a low concentration solution. So it's just called passive because it's not like we're needing to put energy to make it happen. So it's just kind of happening passively. Then we have a channel-mediated passive transport. And this is just a type of diffusion that regulates the flow of ions and molecules through a cell membrane with the aid of channels. So it's kind of like how passive transport, talking about how we said with passive transport, but it's also talking about how now we're using channels to make this happen. Now, in contrast, we have active transport, which is the movement of molecules along a membrane from a region of low to high concentration, and thus we need chemical energy. We need energy to make this happen because it's active. It's having to go kind of like uphill. How that needs more energy than if you're driving downhill or something like that. Then we have a transporter membranes protein, which are just proteins that move molecules and ions along the membrane, just transporting them. Then we have anchor membrane protein, which are these link intercellular structures with the extracellular, intracellular and extracellular structures. Then we have receptor membrane proteins, which are the proteins that relay the signals to the cell's internal and external environments. So the receptors, they receive signals and then they relay it out to the different environments inside and outside the cell. Then we have enzyme membrane proteins, which this is meaning that it just transforms a molecule into another form. Still a molecule, but just different form. Then we have simple diffusion, and that's just the process of moving solutes in a solution along a concentration gradient. And those are transporting and transforming. And then finally, we move on to the last two, which are models. We have sandwich model, which is from Davison and Daniele. And that's the model where it's called sandwich because it's literally a model where the lipid layer is squished between 2 protein layers. So it's kind of like a sandwich. The lipid is the inside of the sandwich. And then we have fluid mosaic model. This one is the one that now it's like we use as theory, and it's from Singer and Nicholson, and that's the model where the phospholipid molecules have a hydrophobic and hydrophilic end, and that makes up most of the membranes. So that one's the main one we go by, and we see all the charts that look like that. And those are our models, and this is my final little mapping of a lot of definitions.